every 8 women will be diagnosed with breast cancer in their life. And it's worse for those with genetic mutations in the BRCA1 and BRCA2 gene. It's estimated that 55 to 65% of women with a mutation in BRCA1 will develop breast cancer before age 70. We have decided to develop a gene therapy solution for those with the BRCA1 gene mutation because these people are estrogen receptor negative, progesterone receptor negative, and HER2/neu negative, which means that they do not respond as well to current cancer therapies. The BRCA1 gene is located on chromosome 17 of the HG38 human genome. This gene encodes a nuclear phosphoprotein that maintains genomic stability and acts as tumor suppressor and produces a protein that repairs DNA. Current cancer therapies for breast cancer are radiotherapy, chemotherapy, and surgery. They all have their risks associated with them. For example, they could affect the surrounding tissues, such as lung and heart tissue, or the tumor cannot be completely removed. Our solution at CarcinoGene employs novel gene editing technologies to create a preventative solution for those with the BRCA1 mutation. Our solution consists of three steps: detect the mutation, create a sequence, and replace the mutation. I will be going through a Benchling simulation in order to demonstrate to you our procedures. Firstly, we're going to spot the mutated sequence through new generation sequencing. There are 3 steps. First, we isolate the BRCA1 gene, and then we amplify it to make multiple copies of it, and then we sequence those copies to find the bases of the allele of our patient. And then we compare the DNA sequence of our patient to known mutated alleles of the BRCA1 gene to find exactly what mutation our patient has. This is all done through computational techniques. In our simulation, I upload our DNA sequence from the NCBI database. Then we're going to create a gRNA, HR template, and plasmid. In this example, we're looking at a patient with a pathogenic variant who has a 1508 base pair long deletion on a part of exon 3 of the BRCA1 gene. This deletion partially removes the C-terminal BRCT domain of the BRCA1 protein, which is important for DNA repair activity. Then we're going to design our single guide RNA. We are using a single guide of a wild-type Cas9 with a PAM sequence of NGG from SP-Cas9 3' side. The best single guide RNA that we can find for this region has an on-target score of 24.9 and an off-target score of 44.3. Optimally, we will aim for an on-target score of 60 and an off-target score of 50. And that is exactly what we do at CarcinoGene to reduce the chances of off-target edits. Next, we're going to be putting this guide RNA and the Cas9 into a lenti-CRISPR V2 plasmid. And then we're going to design an HR template because we're going to repair the double-stranded break with homologous repair. To make the HR template of the 1508 base pair long deletion, we design an HR arm of range 77,789 to 79,297. In this particular HR template, we will be reinserting the correct base pairs before the deletion. The following HR template is now going to be synthesized with de novo synthesis. So once the Cas9 and DNA template have been synthesized through de novo synthesis, they will be delivered to the patient through lentivirus transduction. First, the lentivirus containing the plasmid and the HR template is cultured. Then they are injected in mammary cells in multiple doses. After treatment, an NGS of a blood sample will be taken to measure how efficient the treatment has been and if mutations have been reduced. And then the patient will be monitored for immune responses to the lentivirus, and then the treatment will be repeated every year. We provide a gene therapy solution that will stop BRCA1 type of cancers because we will be preventing the cancer from developing in the first place. Cancer has caused so much trouble for so many people, and it will go on for generations and generations with these mutations, but that can all stop right here.